students, my name is Niyati Said and thanks for watching Edupedia word videos. My topic for the presentation is meiosis, sex and variation. The production of offsprings by sexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes, isn't? Gametes are formed from specialized diploid cells. This specialized kind of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is called as meiosis. Meiosis ensures the production of haploid phase in life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key features of meiosis are first, meiosis involves two sequential cycles of nuclear and cell division called as meiosis 1 and meiosis 2, but only a single cell of DNA replication. Second, meiosis first is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. Meiosis involves pairing of homologous chromosomes and recombination between them. And fourth, 4 haploid cells are formed at the end of meiosis second. Meiotic events can be grouped under the following phases such as meiosis 1 and meiosis 2. Under meiosis 1, there are 4 phases such as Prophase 1, Metaphase 1, Anaphase 1 and Telephase 1. And under meiosis second, there are 4 phases such as Prophase 1, Metaphase second and Anaphase second and Telephase second. Nearly all multicellular organisms, they reproduce sexually by the fusion of egg and sperm. I'm again repeating, like almost every cell in your body, this new cell, a zygote, has a full contingent of 23 pairs of chromosomes. But what about its parent cells, the sperm and the egg? If the egg and the sperm each had 23 chromosome pairs, their union would result in a zygote with 46 pairs, double the usual number, isn't? Theoretically, this cell would then grow into a person with 46 pairs of chromosome per cell. Subsequent generation would have even more chromosome per cell. Now tell me, students, given the length of the human history, can you imagine how many chromosomes our cells would have by now? The answer to this question is, clearly, this is not what actually happens. Even early cell biologists realized that there must be a way to cut in half the number of a chromosome in egg and sperm cells. And to accomplish that task, nature has devised a special kind of cell division, called meiosis. In preparation for meiosis, the chromosomes are copied once, just as for mitosis, but instead of one cell division, there are 2. The result is 4 daughter cells, each containing 23 individual chromosomes rather than 23 pairs. Meiosis is divided into chronological phases just like mitosis, such as Prophase first, Metaphase first, Anaphase first, Telephase first, and then meiosis second, which has Prophase second, Metaphase second, Anaphase second and Telephase second. Now we will be studying meiosis 1 before meiosis second. This is sometimes referred to as reduction division, because it is during the first meiotic division that the chromosome number is reduced from 46 to 23. I repeat that chromosome number is reduced from 46 to 23. Meiosis 1 consists of 4 stages: Prophase 1, Metaphase 1, Anaphase 1 and Telephase 1. Prophase 1. The prophase stage of meiosis 1 is relatively long and can be subdivided into 5 stages: Leptone, Zygotene, Pachytene, Diplotene and Diakinesis. Now comes the Leptone. During Leptone stage, the chromosome become gradually visible under the light microscope. The compaction of the chromosome continues throughout the Leptone. This is followed by the second stage of Prophase 1 called Zygotene. Okay? Each is a thin thread of DNA. Lepto is thin and teen means a ribbon, okay, along which clearly defined beads of local coiling can be seen. The chromosomes, while they have this thread-like form, are called chromatonemata, okay, and the chromosomes appear single because the sister chromatids are still so tightly bound to each other that they cannot be separately seen. During this stage, both telomeres of each chromosome are turned towards and probably attached to the same region of the nuclear envelope. Lepto is also known as leptonema. It is a bouquet stage because all the telomeres tend to contract the nuclear envelope in one spot so that the looped chromosome balloon out from that point like petal. Okay, this is the representation of prophase 1, that is leptotene. Prophase 1 has second stage, that is zygotene. Homologous chromosome, they align directly opposite each other and are held together at several points along their length. Okay. During zygotene, homologous chromosome, they begin to unite, and that is known as synapse by coming into approximate alignment. Okay. Zygo means union, fusing or yoking. Synapses is the process of fusion that occurs between homologous chromosomes, begins at various points along the chromosome and extends outward in a zipper fashion until complete. Okay, when synapses is finished, the fused homologous chromosome, they look like a single chromosome under the light microscope, but they are actually double. Okay. Interface where two homologous unite, that known as synaptonomal, and that can be seen under an electron microscope. Okay. There are two stages in zygotene, that is early zygotene. This is early zygotene and this is late zygotene. In the diagram of early zygotene at the right, the region where the paternal and the maternal homologues have fused is shown in purple. Okay, is shown in purple, and it is showing that paternal and maternal homologues have fused. Okay, in the next diagram, that is late zygotene, both homologous pairs have fused over their entire length, so they are shown entirely in purple. Okay, and zygotene is also known as zygonema. They form tetrads. Once the homolog pairs synapses, they are called tetrads, okay, or bivalent. Bivalent is a preferred term, but tetrad is nevertheless the word more commonly used in most introductory biology classes. Okay, bivalent is the better choice because there are equivalent names for other situations. For example, an unfused homolog is called as a univalent. Three fused homologs, a common situation in plant, is called as trivalent. And then comes a Pachytene stage. In a Pachytene stage, sister chromatids of each chromosome, they separate from each other. This makes the chromosome look thicker. Okay, Pachy means thick in Greek. Homologs are still paired at this point. Pachytene is also known as pachynema. Okay, and crossing over is seen in a Pachytene stage. Crossing over is the exchange of the genetic material between two homologous chromosomes. Crossing over is also an enzyme mediated process, and the enzyme involved is called as recombinase. Crossing over leads to recombination of genetic material on two chromosomes. Recombination between homologous chromosome is completed by the end of Pachytene, leaving the chromosome linked at the sites of the crossing over. Okay, this is the homologous chromosome and the synapse. Okay, and this is the sister chromatids, and these are also sister chromatids, and thus it forms tetrad, and these are the kinetochores of the chromosome. And then it crossing over, that means exchange of the genetic material between the homologous chromosomes. Okay, this red is interfering with the blue one. Okay, and thus red portion has incorporated into this chromosome. Okay, so these are known as crossover chromatids and these are non-crossover chromatids. Okay, Now comes the diplotene stage. At the beginning of this stage, each chromatid of each chromosome is still fused to a chromatid of the chromosome homologue. And as diplotene progresses, these initially fused non-sister chromatin begins to separate from each other. However, they cannot separate directly or completely because they are still connected by two strands of DNA at each of the point where exchanges takes place. Okay. At each such crossover site, the two strands form an X-shaped structure called chiasma. Chiasmata formation is the distinguishing feature of diplotene. As you can see that they have formed X-shaped like structure. Okay, and then comes a diakinesis. During this, the last stage of the prophase phase, the nucleolus disappears. Terminalization reaches completion and the chromosome coil tightly and so becomes shorter and thicker. The nuclear envelope begins to disappear and the centrosome they reaches the pole. As you can see that centrosome has reached the pole, north and the south pole. Okay. And then comes the metaphase 1. In this, the nuclear membrane disappears and the chromosome become aligned on the equatorial plane of the cell where they have become attached to the spindle as in metaphase of the mitosis. As you can see that the chromosomes have become aligned to the equatorial plane of the cell. Okay, where they become attached to the spindle fibers. These are the spindle fibers. Okay. And then comes the anaphase 1. In this, the chromosome now separate to the opposite poles of the cell as the spindle contracts. Notice that in anaphase 1, the sister chromatid remains attached to their centrosome and move together towards the pole. A key difference between mitosis and meiosis is that sister chromatids remain joined after metaphase in meiosis 1. Whereas in mitosis they separate out. Okay, you can see the chromosome movement towards the poles. Okay. And then comes a telophase 1. Each set of the haploid chromosome has now separated completely to opposite ends of the cell, which cleaves into two new daughter gametes, so-called oocytes. A nuclear envelope reforms around each chromosome set. The spindle disappears and the cytokinesis follows. Okay. In animal cells, cytokinesis involves formation of cleavage furrow. Okay, that results in the pinching of the cell into two cells. And many cells that undergo rapid meiosis do not decondense the chromosomes at the end of the telophase 1. Okay, this is the representation of the telophase. And now the meiosis second, and this is essentially similar to the ordinary mitotic division. Each chromosome which exists as a pair of the chromatid become aligned along the center of the cell and then splits, leading to the formation of the two daughter gametes known as spermatids and ova. These are the differences between mitosis and meiosis. Occurrence, if we talk about the occurrence, then in all body cells they include cells, and meiosis they occur only in the reproductive cells. Mitosis is an equatorial division and meiosis is a reductional division. Number of daughter cells produced in mitosis are only 2 and number of daughter cells produced in meiosis are 4. Prophase they involves relatively few changes, whereas meiosis they involve a series of changes in chromosome distinguished into 5 substages, okay, that we have already studied. Synapses and crossing over does not occur in mitosis, whereas it occurs in Pachytene stage of prophase 1 of meiosis. Okay. Centromeres in Anaphase is centromere is split into 2 in mitosis, whereas centromere do not split in metaphase 1. Okay, and Telophase results in the formation of 2 daughter nuclei having the same number of chromosomes as that of parent cell, and in mitosis. And in meiosis, telophase second results in the formation of 2 daughter nuclei each having half the number of chromosomes as that of parent cell. Cytokinesis it follows immediately after karyokinesis in mitosis, whereas in meiosis may or may not occur at the end of the first karyokinesis. I hope it's clear to all of you. So this comes to an end. Thank you and stay tuned and keep watching Edupedia Word video.